name is Mia Crisafoli, Digital Coordinator here at Antibody Engineering and Therapeutics US. With me is Sam Sugerman, Principal Scientist at Fortis Life Sciences. Sam, can you tell me about the advantages and disadvantages of VHH?
Definitely. So we make VHH, which are small single-domain antibodies, where they are expressed in llamas, alpacas, and other camelid animals. And we are able to just isolate the variable domain, where it's about 14 kilodaltons. It's about a tenth of the size of full-length IgG. And it has very unique binding properties, some advantages in thermal stability, and it's able to bind in places and situations where a traditional IgG molecule wouldn't be able to.
Interesting. And what is a problem that VHH would be good at solving? And when would a full-length antibody be better?
Yeah, absolutely. So the full-length IgG is going to have about twice the interface on the binding. And so if you have something that is small or disordered in nature, such as a small molecule or a flexible peptide or a denatured peptide, the full-length IgG is going to be able to form a better interface there. If you have something that is complex and really sort of folded and ordered, then a VHH is going to be able to certainly compete with a full-length IgG. And there are certain circumstances where VHH is going to be better, such as if you want a conformational binder, or if you want to bind to cryptic epitopes, such as the active site of an enzyme that a full-length IgG might not be able to inhibit.
Oh, very interesting. And where do your VHHs come from?
Yeah, we have a herd of llamas and alpacas on site at Fortis, and all of our VHH molecules are naturally derived. So we have a naive library, but sort of our bread and butter are some immune-derived libraries that we do on sort of a custom basis for customers, where we're taking an antigen of interest and we're immunizing it into the animal. And from there, we get a specific and sort of custom library that we're able to apply to any sort of binding property of interest that the customer wants.
Very nice. And how does your phage display support your VHH antibody discovery process?
Absolutely. So the phage display will allow for us to isolate binders with many different properties. And I think that phage display fits really nicely as sort of a complement to other display methods such as yeast or ribosome or mammalian surface display. But phage will allow us to isolate binders for selectivity profiles of interest. So if you want a human-mouse cross-reactive VHH molecule, that's something that phage display is really able to do effectively. But phage is uniquely suited to get a human-only binding profile, where you're sort of cross-adsorbing to a mouse and subtracting the mouse binding VHH molecules from the pool. As well, phage display is really going to help us to do things like temperature sensitivity, where you get a thermal-based dilution, or pH sensitivity, where you're really going to hinder the viability of a yeast cell or the viability of a mammalian cell. And so phage is sort of one of the unique options there.
And where does Fortis fit into the antibody engineering pipeline?
Fortis does a great job at discovering hit molecules, where we are able to find the early binders and hand those off. So we are making research use only molecules. We have very favorable IP. And so discovering early assets for customers is where we really shine.
Amazing. Thank you so much for sitting with me here today. This was such a good conversation.
Perfect. Thanks so much, Mia.